So thank you everyone for joining us. I'm Eric Camino. I'm PPMD's Vice President of Research and Clinical Innovation. I'm joined by a few folks from the Edgewise team. So I have today Dr. Joanne Donovan, who's the Chief Medical Officer, and Abby Bronson, who is their Vice President of Patient Advocacy. And they're joining us today to give an update on both their Becker and Duchenne programs with Sevasemten. So we are recording this webinar. So if you have to step away or if you have friends or family members who this might be relevant to, this will be up on PPMD's website in the following week. So please feel free to share it with them. Um, After we go through the presentation with Edgewise, we will have an open Q&A. And so there's a chat function at the bottom of your Zoom screen. So if you have any questions that come up during the presentation, please feel free to put them in the chat. We'll try to get to as many of these as we can, as well as those of you who submitted questions prior to the webinar. But with that, I think we can start. So Abby, I'm going to hand it over to you.
Great. Thanks, Eric, and thanks PPMD for organizing this opportunity for us to be able to share our latest results. So we can go to the next slide. This is our forward-looking statement. I think everybody is used to these kinds of these slides. I think probably the most important thing to know is that Sevasemten is an investigational agent. It's not approved in any territory. We are intending this presentation for a US audience only. Both Joanne and I are employees of the company. It's a publicly traded company. So those are all the important things. Okay, next slide. So the agenda, what we're going to cover today is we're going to start with a really brief introduction. Then Joanne will go over our Becker data. And we have some new open label data from the MESA open label study. So that's what we'll be going over. And then we'll talk about our Duchenne trials, LYNX and FOX. And then there'll be a Q&A, as Eric said. Next slide. So usually we start these talks with a picture of our offices in Boulder, Colorado, and we say that Edgewise has started in 2017 and is committed to muscle diseases, especially Duchenne and Becker. I think this slide that shows the trials that we've done and started since 2017 really probably visually represents our commitment to Becker and Duchenne better. We started in a phase 1 in Arch in Becker, and using that data, we moved into a phase 2, and also that allowed us to get into Duchenne a little bit earlier. You can go to the next slide. So now I'm going to turn it over to Joanne, who's going to take you through the data.
Okay. Thank you for joining today. We are very pleased to be able to share these data with the community. And thank you to PPMD for giving us the opportunity to do that. So I'm going to start. We have a lot of data to share. So this is really going to be a data-heavy presentation. There's a lot to understand. And we have new data both on Becker as well as Duchenne. So starting off with Becker, as you saw, we have earlier trials in Becker. The Canyon study was a phase 2 multi-center study to assess the effects of Sevasemten on biomarkers. It was not powered for an efficacy endpoint, and it was powered for looking at CK changes, biomarkers of muscle damage, because that's what Sevasemten does. It is intended to protect the muscle from the contraction-induced injury that happens in Becker and Duchenne because of the lack of dystrophin. These were ambulatory men aged 12 to 50 with a dystrophin mutation, a Becker phenotype, not on corticosteroids, and they had to have a North Star ambulatory assessment, that 17 panel scale, between 5 and 32, which is basically in the middle, not at the very top of the scale. And the reason for that in Becker is that that's the group of patients that we have a pretty good idea of how they progress, of how their North Star changes over time. And I'll show that on the next slide. We enrolled 40 adults, 29 adolescents, and the study was over 12 months. The adults were on a dose of 10 milligrams a day, and it was dose finding with the adolescents. And we looked at functional measures, we looked at safety, PK, biomarkers, and the intent was to then be able to develop and to power a pivotal study. This was not intended as a pivotal study. What we found was that the slides don't move. There we go. The biomarkers, as we had seen in a previous trial, decreased as early as a month. The primary endpoint was met, which is great for any clinical trial, 28% decrease in CK, and a 77% decrease in fast skeletal muscle troponin. Sevasemten targets the fast muscle fibers. They're about half of the muscle fibers. They're the ones that get injured early. And it specifically decreases the troponin that's associated with fast skeletal muscle fibers. And I would note that this is different than the cardiac troponin. That's a completely different molecule. So we are getting to the target, and we are seeing decreases in these biomarkers of muscle damage that leak out with the contraction-induced injury. Now, what happens to function? We saw that over the 12 months, we saw that the North Star is stable. The men in this range of North Stars at baseline decline between about 1 and 1.7 points per year. And that is shown on the blue triangle, the natural history. We saw the placebo folks declined at about the same rate as natural history, but the Sevasemten folks were stable over that period of time. Now, what happened after the Canyon study was, this is very encouraging. This helps us then look at longer term studies, the pivotal study that we have ongoing, and we enrolled those men into the MESA study, the open label study. And we actually have 99% of people that have continued from all of the clinical trials that have involved, that have enrolled Becker. And that's our intent, of course, to keep people with access to drug over the long term as we continue to observe safety and durability of the changes in function. So the MESA trial then allows us to look at what happens and with additional time. And what we saw was that with another 6 months in MESA, and there is some variability, but after another 6 months, 18 months, they continue to be stable or slightly improved over that period of time. The men that were initially on placebo, now there's no more placebo after 12 months. And after another 6 months, well, they're stable, even up a tad. And these are meaningfully different than the natural history studies. We also our earlier study was the ARCH study. It was an open label study of 12 men at 2 years. Now they have been enrolled in the open label study for an additional year. And at 3 years now, 11 of 12 are still on Sevasemten. And they're predicted, we developed a model to predict what the North Star decrease would be depending on the baseline function in their age, their baseline characteristics. And they are stable after 3 years, which the longer you go, the more information you have when it seems to be diverging from natural history. And they would have been predicted to go down by about 4 points. So we're very encouraged by that. And that allows us, of course, that had allowed us to power a pivotal study. And I know that, for Duchenne, generally the decrease is greater, but in Becker, 1 point differences continued over 10 years, over 20 years. is really important. And even a 1-point difference, these are the kind of things that 1 point on a North Star is for a patient with Becker, to needing assistance to go up a stair, up a curb, to being able to use the toilet independently or not, or to be able to get up from a fall with assistance, or even to get up at all. So we think these are clinically meaningful. This allowed us to power the pivotal Grand Canyon study. It's fully enrolled with 175 patients, 18 months, similar population, adults not taking corticosteroids, and it's well powered. That study is on track to have top-line data towards the end of next year. And at that point, we would then take it to the FDA and file it, and then it's in the FDA hands to go through the approval process. So that's where we are with Becker. To go on to the Duchenne studies, we have 2 Duchenne studies in children with Duchenne. The LYNX study, which is in children who are largely on corticosteroids. And I just want to back up one step with the mechanism of action here. That because what the drug is doing is inhibiting the fast muscle myosin. It's the contractile protein. So in a mouse, if even a few contractions can cause visible injury in the mouse, and I think that you've probably all seen this video. If it's contracting at 85% with Sevasemten, then function is preserved, but there is protection, that you're not seeing these blebs, which are really rupture of the individual muscle fibers after influx of calcium and a whole series of effects that contraction causes to induce injury in the fast, particularly in the fast muscle fibers. So our goal is, as it was in ARCH, to find that dose where we're able to protect the muscle, but not inhibit it too much. So that's why we went through a fairly extensive dose finding selection in Duchenne. We did that in the earliest studies in Becker as well. We went up in dose, we found the ideal dose, and we came back and targeted that dose in adults with Becker. So our goal was, and how we were designing these studies, how we were thinking about them. What we wanted to do with these phase 2 studies was to look at safety, of course, first thing, and to identify a potentially beneficial dose in phase 3. They're not designed to be efficacy studies, but they are, do we have a signal there to move into phase 3? That's the goal of this. And that will then give us information about, well, what patient population would we look at in the phase 3 study? What's the background therapy? Our goal is to develop a treatment that would be able to be used across different treatment backgrounds. So a foundational therapy, basically, whether it's on top of steroids, on top of dystrophin-targeted therapies, or other mutation-agnostic therapies. And the way we designed that, we looked at how we had seen the biomarkers fall quickly within a month in the patients with Becker. And what we designed was, in an effort to decrease the length of the placebo control period, we designed a 3-month period to look at biomarkers for dose selection and potentially be able to determine dose on that basis. And then we had a much longer open label period where we would continue to evaluate function. And what we were planning to do then in that open label period, if we didn't see effects in that first 3 months, then we would go up in dose. And in fact, the FDA encouraged us to do that, because what we don't want to do is leave boys on a dose that is not beneficial. So we want to, this is an adaptive design, but we want to react to the information and change the doses in the study as we are seeing data. So the LYNX study, two-part, as I said, multi-center in the United States, randomized placebo-controlled study. We looked at 5 different doses, and we also looked at a small number of boys who were not on corticosteroid therapy. They were steroid naive. And the primary endpoint was safety. These were 4 to 9-year-old boys, ambulatory. In general, they needed to be on that stable dose of steroids with less than 10-second rise from the floor and 10-second 4-stair climb. And after the initial period, we had the open label period, which is now out to 92 weeks with dose escalation. And as we'll see, we actually then came down in doses. So this is the demographics, the background of these patients. And we enrolled 66 patients. I'm showing you cohorts 2 and 3 because those cohorts ended up largely on a dose of 10 milligrams. And spoiler alert, that's the dose that we ultimately decided was the dose that we would move on to phase 3. So I'm showing you those folks individually. And while the age at enrollment was between 4 and 9, clearly this is tilted towards the higher ages. So the boys were almost 8 at the beginning of the study. And over the 18 months of data that I will show you, now they're on average, they're over 9 years old. So these are a bit older. They're certainly older than the cohorts that you've seen in gene therapy studies, in most of them at least. And these are the baseline values of their functional tests. 60 of the 66 were on a stable steroid background, and 9 of 66, so about 15%, were on an approved exon skipper. The boys that were not on steroid, the 6, could move over onto steroids as of 16 weeks into the study, so that the intent was not to have them long-term not on steroids, although a number of them actually have stayed on just Sevasemten and not moved on to steroids. But that's per the parents' choice. That is not through via the protocol. So in terms of safety, what we looked at the placebo-controlled period, the 12 weeks, and we looked at these with the Data Safety Monitoring Board composed of multiple neuromuscular docs, as well as a patient representative. And we saw that this is the highest dose, cohort 5, cohort 4, the 15 and 30 milligram dose. And we saw adverse events, side effects that were similar to what we had seen in the Becker patients. Dizziness, somnolence were the most common. They tended to be mild and transient, usually at the beginning of dosing, and then so it's when the dose is changing or when the amount of drug in the body is changing, they tend to, and then they level off, and then they with time, they tend to not be an issue. And in fact, that's what we saw in the Becker study, that even at the highest doses, once you're stable, you do not have those side effects. When we moved, and this was why we stepped up gradually, basically, in the dosing by looking at the previous dose and going up. In general, it was similar in the open label period, but as I'll go into more in the FOX, what we started to see was that they were basically at the highest doses, the boys were slowing down. And the parents told us that in the boys in the gene therapy trial, in the trial that was in boys that had already received gene therapy as well as here. And so how are we making these decisions in terms of doses? Well, as I told you, we initially focused on the biomarker changes at 3 months, and we didn't see CK changes, which surprised us. We saw TNNI2, the fast skeletal muscle troponin changes, and we saw it at the highest doses. There was other a proteomic panel that we did see moving at the higher doses, but we didn't see characteristically what we had seen in the Becker patients at these doses. But what we did see actually was in the cohort of patients that were steroid naive, that's this cohort, compared to the cohort that was on steroids at the same dose. And this is the second cohort, the 5 milligrams. And there we did see a difference in CK. We did see a difference in the fast skeletal muscle troponin. So that, that opens the possibility that is the steroid somehow stabilizing the membrane and we're not seeing that biomarker change. And we noted that actually in Becker, where they weren't on steroids, we did see these changes. So is that, and I will say we don't entirely understand this at this point. I can't give you a clear rationale, but that's what we are thinking. And we shifted to functional measures to understand what's going on here with the dose, because clearly something's going on at the lower doses. And we looked to SV95. We measured North Star. You are very familiar with North Star, very well accepted, recommended. It identifies changes in everyday life for these boys, and it's used a lot even in clinical practice. The stride velocity 95th centile is a newer measure that is basically done by the boys wearing an ankle sensor every day for 3 weeks. So they do this for 3 weeks before they come in to visit. And what that does is tells us the analysis tells us the maximum or near maximum speed that the boys are running at. And the boys will run as fast as they can. So this turns out to correlate very well with North Star. It correlates well with other patient-reported outcomes as well as other functional measures. And in fact, the European Medicines Agency, their FDA, has approved it for use in trials as a primary endpoint. So the advantage of it is that it seems to be more sensitive even right away within the first 3 months than the traditional North Star. And of course, it takes advantage that you're looking at something that's reflecting everyday life. It's not just in the clinic. And we know the whole community has struggled with that and with different measures evolving as trials advance, and what's the best measure to use and trying to avoid its variability. So what we found with the stride velocity is that when this is the change in stride velocity, so the dotted line is no change at 3 months. And what we saw was that the placebo and the doses up to 10 milligrams looked okay. They were basically no different from zero, from placebo. Now, the placebo stops after 3 months, so we don't have any data after 3 months in the placebo, and then they go on to Sevasemten. What we saw was that the boys in the 15 group had slower stride velocities, as well as the 30 milligram group, which was what we were hearing in the 30 milligram from parents via the investigators. So not everybody, but some. And what we did was that we dialed that back. We moved to lower doses, and we settled on doses of 5 and 10. as I told you, this was a complex study. We are looking at the data. We are figuring out what the best thing to do is next. And that was, and as the people who were in the study know, we did move the doses around as we learned more during the study. So we moved everybody to either 5 or 10 milligrams to understand what's happening over the longer term and potentially to identify a dose then for phase 3. So I'm going to take you through complicated data here. And so deep breath. This is just stepping through. This is cohort 1. Those folks started out on up to 5 milligrams, and we dosed them up to 15 milligrams as we saw the safety, as I showed you in the first 12 weeks of once the 15s were the 15 information. This is the absolute SV95, the change, the stride velocity, the change in that over time. And what we saw for these guys was that they were generally doing better. The gray line is natural history. They were generally doing better. Then they did worse when we went on the 15, and now off the 15, they're back to 5. They're doing better. What we saw in this group, which is mostly on the 10 milligram dose, and these are relatively small numbers. I should have mentioned that back here, these are single-digit numbers. We have small groups of boys, but are able with this to be able to see these differences. The boys then move over onto the ones who were on placebo, move to active. So here we have 23. And after a year and a half of 8-year-old, almost 8-year-old to almost 9.5-year-old boys here, they are stable over that period of time. They went down a bit when they were on the 15, but they are stable and they improved over that time. And so this is what you would have expected the natural history to be from the data in the literature. And that's really encouraging. These are the 95% confidence intervals, which means this is quite a bit a ways from the natural history in terms of what we would have expected. The cohorts that 4 and 5 were worse. They felt good. We didn't hear any adverse events in here. And after they moved on to the 10 milligrams, they're a little bit earlier, so we don't have 18 months of data there. They got better, and they got faster, and they're actually above where natural history is before. So I'm sure you will have questions, and I'll come back to this when we do. But basically, we settled on this is quite promising to have this stable over 18 months. In relatively older boys. What happened to other things? So the North Star over the 18 months for the cohorts 2 and 3, largely on 10 milligrams, went down 1.5 points. So that's about 1 point a year for almost 8-year-olds. And what we did to compare that, because we didn't have a placebo group, is we compared it to a multivariate regression model that Professor Montoni developed with his colleagues based on their function at baseline, age, height, and weight. And that would have predicted they would have gone down about 3.5 points. So a couple points better than predicted. Now, this isn't a direct comparison, but we're trying to get a sense of versus natural history here, what would have happened with these boys. The SV95 was pretty stable compared to what it would have been, what natural history would have predicted. And the 4-stair climb was also stable with a mean change from baseline of about 0.5 seconds. So there isn't a good comparison there. So that's where we are with LYNX. We have most of the data, certainly in the boys who have not previously been treated with gene therapy. We started the FOX trial to understand what would happen in boys that had been previously treated with gene therapy. And that trial was designed initially with 2 doses that at that point were doses that had been shown to be safe in the placebo control period, open label extension. And these were ambulatory boys and adolescents aged 6 to 14. They needed to be 2 years after gene therapy. So these were all boys that had been treated in the clinical trials. They had to be on a stable dose of steroids, either daily or weekend was fine. We enrolled 32 boys. 2/3 of them had been in the Elevidys clinical trials, and a quarter had been in a Pfizer clinical trial and some in the solid clinical trial. So how do we compare these? What do we compare to? Because there isn't good natural history that's available, and that's going to be a question of how to do that. So in terms of what do these guys look like, they're older. These are 11-year-old boys at baseline. So the natural history is if they hadn't been treated on gene therapy, of course, they would be clearly in the stage where they're going down in their North Star. And the boys who had been treated with Elevidys, they were also almost 10 and a half. They were also 4 years out from gene therapy. So what we looked at was there is some data in the literature between maybe years 2 and years 3, but first safety. So here at the 15 and the 30, these are older kids. And we did see more, we did see that there was more dizziness, more somnolence. These boys at 30 milligrams were reduced to 10 milligrams within the first 3 months, and subsequently in the 15 milligram group was reduced to 10. So they've been on 10. They haven't been on 10 as long as the boys in LYNX. So that also, it's a complicated study, basically. What we do know, the only data we could find really that was out more than, in a significant number of boys out more than 2 years was from some of the posters on Elevidys. And between year baseline, basically to 2 years out, the boys on Elevidys were up on their North Star, 2.6 points. There's another poster, this was 63 boys. There's another poster with 50 boys that were out 3 years, and now they're down about 1.36 points. So they seem to be going down in their second year. Not all of the limitations, all the caveats here. These aren't exactly the same boys. We don't have information. Nobody has this information. The rise from floor at the end of 2 years was almost stable. At the end of 3 years, that group was up about 4 seconds. And similarly, the 10-meter walk was up. Now, those kids at the beginning of this, between 2 years and 3 years, are about 8.5 at that time. Our kids in FOX, now they're further out, they're over 10 years. So what we saw was, and this is not far from a perfect comparison, but we saw they were going down with a North Star of 2.5. Their median change in rise from floor and 10-meter walk was within a second. So that's encouraging. But we really need to look to the LYNX data to how are we going to choose a dose here. So what are we going to do next? We are going to continue dosing in these studies in LYNX and in FOX. We are preparing for a phase 3 study. We believe that these observations support the the 10 milligram dose. That was the intent of the study, to look at safety, to look at function and be able to, and biomarkers, and be able to choose a dose. What are we going to do? We know that there are boys on the 5 milligram dose who have come into the study because we were focused on the 5 and the 10. And what we are finishing up is looking at the blood levels of Sevasemten, and we will to determine what the appropriate dose adjustment would be. We think that we're going to move most, if not all, of those boys up to the 10 milligram dose, and that will happen very fast within the next month or so. We're going to look at the primary and the secondary endpoints and try to sort out with regulators, with FDA, with EMA, what makes sense for a phase 3 study. Are they in agreement with this? Are they in agreement with looking at boys not only on the background of steroids and exon skippers, but also potentially on a background of gene therapy? How do we adjust that population? Because again, we want to develop something for all of the boys and young men. And things, obviously, things are changing all the time in this. So we've got to weigh this against the therapeutic landscape. And so what we're doing is we're preparing a package to go to the FDA. And of course, that takes a few months to go to the FDA onto those. So we anticipate we'll be there in the fourth quarter of this year, and then be able to come back with more information on what the phase 3 would look like. But given a discussion with the FDA towards the end of the year, we are looking at 2026 for a phase 3 study based on all of this information that we're going to be continuing to gather. That's a lot. I've told you probably a lot more than we usually tell, kind of gone into more detail. So I'm sure you'll have questions. And I can kind of see that there's a lot of questions in there, but I haven't had a chance to look at that yet. And Eric will be good about that.
Yep.
And I'll just let Abby sum up.
Yeah, that was a lot of really good information, Joanne, but I think it was important for everybody to hear how, what's going on in Becker, and then how we walk through the Duchenne story. So in summary, we are very pleased with our progress, and Sevasemten, as you just heard, it continues to show promise in both indications. So in Becker, we're continuing Sevasemten dosing. We have the Grand Canyon, that's our pivotal trial that's on track for reading out in the fourth quarter of 26. And we, recent regulatory engagement, we have a clear path to approval with a primary endpoint, and that is the Grand Canyon trial. So we feel really good about that. And in Duchenne, as you just heard, we saw that Sevasemten was associated with reduced functional decline. It's a complicated study, as Joanne said. We're figuring out a path to phase 3 and have decided on 10 mg as the target dose. And we're planning to meet with the regulators in the second half of 25, and that will help us refine our plan along with everything. As Joanne said, things change, and we just want to always be making the right decisions and trying to best meet the community's needs and figure out what's going on. So that's sort of the wrap up. And now I think we have some questions. And Eric, do you want to take a stab at this?
And lastly,
yes,
we want to thank.
Yes. I'm sorry.
This has been a long study for people, and we really appreciate the families and the boys and the men in the Becker study who have been coming to clinic over this period of time. So we realize the burden that is on folks, and appreciate your commitment, your dedication, all your efforts. So thank you.
Yeah, and thank you for the presentation. That was a lot. We do have a lot of questions coming in. So I think, and we had a lot submitted beforehand as well. So try to get through as many of these as we can. If we could maybe go all the way back up, I think some of this will be easier if we kind of go through some of the slides and where some of these questions were kind of asked and maybe relevant. So maybe back to slide 5. And just
Okay.
as a start, just a, softball to get us started. Across both Duchenne and Becker, are there any mutation restrictions for Sevasemten?
No.
Okay. Thank you. Just want to make sure,
Okay.
clarifying for everyone that this is not the
Mhm.
case of
Yeah.
those we do have sometimes.
There, there are, in Becker, there are some mild mutations. We're not selecting on that basis, but when we have a baseline North Star between 5 and 32, what that does is basically screens that we don't include the people who have a very mild form of Becker. So.
Yeah, and that was going to be kind of my first question was that there was a question around not including Becker patients with scores of 33 or 34. And I know
Mhm.
we've talked about this previously as to why, but maybe you could explain a little bit again in terms of a clinical trial design piece, why
Yeah.
that's relevant, and then whether
Yeah.
or not that is a potential benefit for patients with those still high
Hmm.
scores.
Yeah. So there are good natural history studies, and when you look at and actually Luca Bello of Italy first identified this as a way of running studies in Becker, and the more recent studies in Becker have used this kind of exclusion, but once people start to decline, they seem to decline fairly reproducibly. I know you've seen the graphs of in Duchenne that there's, at different ages, people peak at different ages. But in Becker, it does seem that once you start to go down, once you start to lose the North Star, then you continue to go down. And so that's why we look at this group, because in a clinical trial, what we need to look at a group of patients that is going to change over time so that then we can measure the effect of change. We can measure the effect of the drug on that change. So if we're looking at people who aren't going to change over time, and with the North Star, they're already at the top, they can't get better, then that isn't going to be able to help us understand if the drug works. And so what we want to do is we want to enroll, and you've seen this in many trials in Duchenne, you want to enroll a group of patients that is similar enough that there isn't a lot of variability, because the more variability you have, the less powerful a study is to be able to look at a drug effect.
Perfect. Thank you. Um, and then since we're on this slide and talking about adults with Becker, we did have a question of, is there been any work to look at potential impact on reproduction given this kind of our adult cohorts? So this doesn't come up as much as sometimes in our Duchenne studies, but I think
But
it's an important
yes,
question.
we have done all of those reproductive toxicology studies, which is what you need to know whether or not to allow men to father children or not. So we have done those, and we are in the process of updating our guidance in the protocol, and we have allowances there. So and that's something to discuss with the investigator, but we are well aware of that. That gentlemen in their 30s, yes, these are people with families. They want to have families, and that was a consideration to make sure that we got that understanding of that early.
Great. And then on the next slide,
Yeah, excellent. Okay.
So there, question was in terms of, and did you break out, I guess, in between, for all of these, I guess, the level of granularity of changes in some of these biomarkers based on patients starting North Star scores? So were there bigger changes based on having better function at the start or maybe more progressive disease?
Yeah. So there are 28 patients on Sevasemten here. And so we don't have, there aren't obvious changes based on baseline. One of the exciting things actually about having 175 patients in the larger study is we'll be able to look at a lot more of these correlations between biomarkers and function. So
Yeah. And I think this is part of the challenge as well when we get into some of the Duchenne data with just the smaller numbers. It helps point you in the right direction, but is a bit of a limitation. On slide 7, so one, I think just helpful to acknowledge, you showed here both that there is this natural history cohort, as well as you have this placebo, and there is kind of this nice alignment in terms of their progression to what we would maybe expect based on this data. I know it sometimes comes up of how contemporary is our natural history data. So
mhm.
I think it's just
Yeah.
for people that are watching, it's important to acknowledge as you kind of continue to show data
Yeah.
predictive. Um,
It's different in Becker. It's very recent, actually. There's a couple papers from 24 and I think 22 are in here. One that is the in poster format only right now. Also the standard of care in Becker hasn't changed. So it is a little bit more reliable, I think, to look at natural history there as well.
And then there was a question with regard to, so when talking about the North Star in Becker and looking at, what is pretty stable or sometimes these small gains in function, have you broken down and looked, is it specific tasks within that kind of 17 item task list that are tending to change versus other ones?
Yeah, we have again with small numbers, it's hard to say. I think stepping up and down tends to be the one that changes the most. So,
And then on the next slide, um, again, I think, oh yeah, the note again of, can we just, I had a question, there was about just reiterating the timeline for submission to the FDA, and then would be under the traditional pathway or an accelerated pathway?
yeah, I don't know if you're gonna, so the Grand Canyon is going to finish in the fourth, or we'll have top line data. We asked the FDA whether or not we could go with an accelerated. Now, we don't have dystrophin. We're not doing muscle biopsies in any of these. We don't anticipate dystrophin would change. And we have biomarkers, but the FDA isn't completely comfortable with using things like CK and TNNI2, fast skeletal muscle troponin. What they said is, we like North Star. It's a great endpoint. It's not an accelerated endpoint. It's a full endpoint. So come back to us. We like the Grand Canyon study. And so we have talked to them. We're going to continue to talk to them also about the modeling of the natural history data, because we think that can be very helpful as well.
And then a question we had as well is, so all of these studies are adult Becker patients. Will there be any opportunity for pediatric Becker
Yeah.
patients?
Mm-hmm. Yeah, we have enrolled adolescents. We'll be sharing that data at an upcoming medical meeting. And we have, so we believe that it is going to be, we will look to a broader label. That depends. But I think one of the things that we do have is we have experience. What the FDA and EMA want to see is, do you have safety across the age groups? Do you have which we do because of the Duchenne boys as well. And are we able to look at PK, the exposures? Do we have the right dose? Those are usually the two major things that they look at. So that is we are looking to ask, the FDA has historically given an age beyond what is just in the clinical trials.
Okay, yep.
So.
All right. If we move to maybe slide 15, if I have my slides correctly. Yeah, okay, great. I think that, so you talked a little bit here about kind of the dosing and the potential for inhibiting kind of muscle function. Is this, as you were looking at those higher doses in the Duchenne study, is that some of the thought that some of that is mechanism of action related? Just maybe
Yeah.
explaining here is because this is, I think, one of the best visuals for people to understand, like, what does Sevasemten do?
What is it doing? Exactly. So we don't want to, we're inhibiting that ATPase that controls contraction. And we don't want to inhibit it 100%. We want to be as we want to protect the muscle, but without changing function. So if you go to higher doses, then the expected pharmacology is to slow things down as well. So that's exactly. Now, the good thing is, having gone to those higher doses, we don't see cardiac. The molecule actually doesn't interact with cardiac contraction at all, cardiac myosin. We don't see cardiac. We don't see kidney adverse events. We don't see liver adverse events. So even at those doses, but we do see more pharmacology than we want, basically.
Yeah. No, that's helpful. I think as families look at some of that data you presented, I think that's just helpful
Mm-hmm.
to have back of mind.
Mm-hmm.
And then maybe to slide 18, which I think was some of the demographic. Yeah,
Mm-hmm.
so here are just a couple of things to touch on. So the stable steroid background, is that prednisone, deflazacort, and vamorolone, or just two of the
We
three?
allowed vamorolone in the open label period. We did allow boys to move over to vamorolone as an approved steroid.
And then you marked here as well that the exon skipping drugs were studied. Any opportunity for a Gamry, sorry, not a Gamry, that is, this is when I'm using the wrong one, Duvisat, Givinostat.
That. Yeah, we haven't included Givinostat, and I know that is an active question. We've spoken to a fair number of the investigators and other experts, and it will prevent us. We won't be able to see what's happening with Sevasemten, and that's why we have said no. And what, there's a couple patients that have dropped out to go on Givinostat. They're certainly free to do that, but that would complicate the interpretation of the information. So, and that's where we are still collecting data and trying to understand the longer term. That now in the future, can we do that? Can we include patients in the open label that are on Givinostat? And again, we hope to, but it is complicated, and that's something that we have asked people and gotten feedback on from docs.
Yeah, thank you. I mean, that's helpful. I think that's a big one in the community. So it was nice to see the inclusion of most of the approved therapies there. And again, when we get to the FOX study, starting to gather some of that data just to help, if there is an approval, to help in the insurance and payer
Yeah,
landscape. So if we move to, so I'm also just don't want to, if we stay a little bit over as we get through questions. Okay, good.
fine. Yeah,
Perfect. So slide 21.
nothing more important than explaining this. There's so many questions. 21. Yeah.
Yeah, so I think here, just in terms of showing kind of these patients that were steroid naive versus patients that were on steroid and the masking idea, but the values here, this is showing individual change from baseline, not kind of raw value that uh, correct that it's not like not that there's a substantial drop in CK or TNNI2 if you're not on steroids compared to if you are,
That's
but
right.
it
This
is
is compared the
individual.
beginning of the study and 3 months later when they've started Sevasemten. Yes, we're
Yeah, just
seeing this change.
thank
Exactly.
you. Just wanted to make sure we kind of clarified that in here.
And steroids do drop CK by about 30%. So we're digging into this. So
Yeah, because I think it's a question of, right, are these patients on steroids also just at a lower number already when you're looking at these, if we looked at
yeah,
the values.
yeah,
Um, and then I think on slide 23, so so one,
yeah.
I think is just a good, and you did a really nice job of, I think, touching on this, Joanne, of just a reminder for families that these kind of early phase one, phase two studies, this is part of the goal of these, is that to identify what is the appropriate dose to move forward in your pivotal study. So I think it's helpful to see just kind of what this can look like at times, and also speaks to some of the challenges in translating therapies in Duchenne, or, even when you have predictive preclinical models, or when you were looking across at maybe some of the data from the Becker, and how this can be challenging in terms of just translation in general. There was a question around, now it was a very small number that you had of patients that were on exon skipping therapies. Were there any notable differences in those handful of patients on exon skipping therapies compared to not?
They were not evenly distributed across the cohorts, though. So they were actually fewer of them in the cohorts 2 and 3. So
thanks. And then the next slide, which has kind of all of the different cohort data and the showing as well, kind of the higher dose.
yeah.
Yeah. So, and I don't know if we maybe click through this to when it has all of the cohorts on.
Yeah.
Okay.
Yeah.
So I think one, just noting that, so in most of the instances when the patients came back off of this high dose, we see that there is kind of a rebound back to baseline that we're seeing in most instances, or towards baseline, I should say.
Yeah. Yeah, we did see that in the different groups. And the so that it is a very consistent finding when they come off the 15 dose. And as I said, they felt good on the 15 dose, but we also don't want them slowing down. So, and what we'll look at is the pharmacokinetics of the doses, and also look at that with respect to the 5 milligram dose, and bring boys up as appropriate. Now, is there an intermediate dose there? We're going to look at the PK carefully and see, is there merit to 7.5 with some boys or not? The idea is, we want to get things right. It may not be one size fits all. And you remember, we've got the 10 milligram dose in the adults with Becker, and we've got 10 milligrams. So that sounds like a lot. The drug goes into muscle. It's 100 times more concentrated in muscle than it is in the bloodstream. And when we look at how the drug is distributed in the body and how it's eliminated, it turns out that the more muscle you have, the longer the drug stays around. And that's kind of why that I can't go through quite all of the steps, but that's why it ends up that it's the same dose in adults and kids, actually, because the kids eliminate it much faster. That's why they need a relatively higher dose than the adults based on their weight. So
that's helpful.
A lot of our drugs, right, are based on weight. They're mg per kg, and this is not.
Yes, because we did have two questions coming in of if there was, heavier patients, would there be an opportunity to start at a higher dose and maybe try to titrate down?
Yeah.
There was also a question around, and you touched on a little bit of, that 2.5 kind of step between the 5 mg and the 10 is the 7.5, finding that sweet spot.
Yeah.
And then from here, so some of these patients were on 5, some were on 10, and you said at the moment, not all patients have been moved over to 10 milligrams.
Yeah, all of these guys, 2 through 5, are on 10. There are a couple that the investigator has moved down to 5, and that's up to the investigator if they want to go back up to 10. But the ones who are on 5 and we started some additional cohorts that aren't shown here. We started some additional boys in cohort 2 that are earlier or on 5. And we're going to look at the plasma levels in boys and give a final answer on moving those kids up in the very near future.
Great. All right, and then if we move to, I think slide 27, so moving into the FOX study.
I've lost my biomarkers.
I think maybe back one.
27.
Yes. Perfect.
29. Yes.
So I think one, you did a nice job, and I appreciate acknowledging some of the limitations here. And I think it is really important just to reiterate that in a study like this, we have a number of patients who their background gene therapy is different across these. And we know that we've had different outcomes in terms of these trials as well. And you talked about many of them, they're a number of years out from gene therapy, and we still are working to understand kind of what does the natural history look like for a patient that has been treated with a gene
Mhm.
therapy. And again, challenges within the different gene therapies
Yeah,
that have been delivered. So it
yeah.
makes this, it's challenging, but I do think as well, we do want to, acknowledge and appreciate that, the families who have received gene therapy have really strongly voiced their want to continue to find other therapies for the
Mhm.
boys and often feel excluded from participation in clinical trials. So we do
Mhm.
appreciate the conduct of the trial. Uh,
Mhm.
so within, I think maybe slide 29. Oh no, we're on slide 29. I'm trying to look at my notes as I scratch these down. Maybe it was slide 31. Uh,
Mhm.
so just the doses here, just reflecting. So higher doses than what were kind of done in the previous study that we just talked
Yeah.
about. So and
Well, we did have the 15 and 30 in
oh,
the LYNX
yes,
study.
yeah.
Yes.
But those were our doses where when you showed that was kind of where it was potentially too high of a dose, and we saw some of
Hmm.
the decline. And then
Hmm.
you said since then they've all been moved to the 10 milligram.
Yeah. Yeah. These were even moved earlier than the 12-week period. I will say I didn't show the SV95. We don't have all of that. That takes longer. We don't have them all for 12 months. So
Okay.
this is, and again, that points out this is interim data. These are not final data. This is as we are moving along here.
And
So
then on these doses, were the patients at, was the length of time that they were on the 15 or 30 mg variable within the study before they were shifted to the 10 as well?
it was because they were enrolled over a period of time, and then we said, no, we're going to move you down. So they were somewhat variable. Again, it makes it hard to interpret it.
Hmm.
I think that's why we're just kind of doing it at a very high level here for the boys in Fox.
Yeah.
So I think that when we consider the boys are 11 years old, basically, in this study, they seem to be relatively stable. Now, again, we don't know the natural history, so we have to take that
Hmm.
caveat into account.
Yeah, and I think that was a little bit on slide 33 where you have some of the data
Mhm.
here. So I think this is like a key point, and I appreciate it. You kind of talked through this as well, that some of the challenge here is that, the Fox study has not just patients who were treated with Elevidys. There's also some
Mhm.
Pfizer and solid
Yeah.
drugs. These are also data in terms of performance based on some poster data that was shared. And
Yeah.
so it's hard, and there could be some of these patients, there could be a chance there's some overlap. It's also hard to
I
know.
bet they are.
Right.
Yeah, I bet these guys were in these ones as well. So
Yeah.
because this is just the Elevidus kids.
Hmm. And so I think too, so I think a good thing here that we're seeing is potentially from a safety standpoint, that you were not seeing any, um, and I know you have the safety data on the other piece, but there no potential um, indication of a safety risk of treating
Hmm.
Sevasemten to a patient who had
Yeah.
received gene therapy sometime
Yeah.
after. And that also, these are in older patients. You're seeing similar to what you showed in the other Duchenne trials. There is this slowing generally.
Mhm.
So again, it's hard to, how much weight we can give to the small
Yeah.
numbers and the variability, but I think these are encouraging results. And I did just want to stop there for families who look at this, just there's a little bit of that context as they
Yeah.
try
It
to
needs
make a personal
context.
decision. Yeah.
Yeah. I I didn't notice people asked about cardiac. We looked at echoes as a safety measure. We talked with folks, the usual folks that talk to you all the time about what we should be using. And we looked at that every 6 months. And we did not see changes in either the larger number of Becker folks or that. There was no negative effect. They were stable over time.
Great, thank you. Yeah, no, that did come up a few times in terms of looking at that. We also had some questions about not just cardiac MRI, but are you looking at it all with either in the Becker or the Duchenne population, using MRI to look at skeletal muscle?
And we are in the Becker population. We don't have, it was a subset of that small subset that had MRI in the Canyon study. We have almost the vast majority of people in the Grand Canyon study have MRI, and we're working with Imaging NMD on that. They've got a lot of good data on Becker as well to compare to.
Yeah. And then I don't know if we want to go to maybe the next slide after that had some of the summary pieces. So one of the questions I think in we had come across a few times was potential for females to be involved, so female carriers to be involved. Is there potential that Sevasemten could be protective for them as well? And then any opportunities or plans for studying that population?
Yeah. I that looking at females, I mean, they are essentially Becker, right? They have decreased but still present amounts of dystrophin. So should the drug work there? If it works in Becker, I think so. We also are looking at certain limb girdle muscular dystrophies where contraction-induced injury is important, and we've done some pilot studies there as well. So we think it could be used beyond that. One of the hard things about females with dystrophinopathy, and I know PPMD is working to fix this, is what's the natural history? Because again, you need to have an idea of the natural history to be able to power a study. So.
Yeah. Well, I know we ran over a little bit, but I do appreciate you hanging on, both Joanne and Abby, to go through. This is a lot of data. And again, this was all recorded so folks can take another look at it afterwards. And if you have additional questions, please send them on to us. We will pass them along to the Edgewise team and try to get those answered for you. But I think we could probably wrap up here. And so thank you both for joining and presenting some of this data today.
Thank you. Thank you
for having us.
Yes, really appreciate all your support for the community and for moving things forward. So thank you. And thank you to everybody.